In this video, we will learn how to build and simulate a Synaptome model in the OpenBrain platform. A synaptome is a neuron model along with its synapse sets. Let's start. On the home page, click on Go to Virtual Lab button to go to your virtual lab. Click on Projects on the left menu. You will see the list of projects in your lab. Make sure to create one if you don't have any projects listed yet. You should also have credits in your projects. In case you don't, then you have to transfer them from your virtual lab to your project so you can run simulations. Click on the Go to Project button on the project that you want to work on. On your project homepage, click on the Build button in the left panel. Next, click on the Build button in the Synaptome tile. For this experiment, we will use a rat somatosensory cortex layer 5 pyramidal neuron model. Now, give a name and a description for your Synaptome model and press Start Building. Select the model and press the Use Single Neuron model to load it. Now we can start adding synapse sets to the neuron. Let's add an excitatory synapse set on the apical dendrites. Give a name, select apical dendrite as targets, and excitatory synapse type. Now we can distribute the synapses using a mathematical formula for number of synapses per micrometer of distance from the soma. We use 0.003 times x synapses, where x is the distance from the soma in micrometer. So for each apical dendrite section, distance of 1000 micrometer, there will be 3 synapses distributed along different branches of the apical dendrite. The synapses are distributed randomly using a seed value on the top right corner of the page. Here we exclude synapses with a distance less than 50 micrometers. Let's add another synapse set for inhibitory synapses on the basal dendrites, but with a sparser distribution. Select basal dendrite as target, type inhibitory, and add a distribution formula. Apply the changes and save the synaptome. We have now built a synaptome model. You can also save it in your library to revisit the model. Now let's simulate it. Go to Project Homepage, click on the Experiment column in the left panel. Next, select the Synaptome tile. You will see a list of pre-created Synaptome models along with the one you created at the top of the list. Select your Synaptome model and press the New Simulation button that appears on the screen. Enter the experimental parameters. In the Synaptic Inputs tab, you can define synaptic parameters, such as synaptic weight and input frequency, for each synapse set that we just created. You can add the second synapse set using the Add Synapse Input button and then selecting the other one for basal dendrites. In the Stimulation Protocol tab, you can add a current injection stimulus and set its parameters. You can add multiple recording locations by using the Add Recording button and selecting the neuron section. You can also do it visually by clicking on any neuron section and pressing the Add Recording button. Once the stimulation and recordings are configured, press the Simulate button to run the experiment. You can interact with the traces and save the traces for future reference. The simulation configuration and results are saved with the Synaptome model. You can also save the simulation recordings to your library. In the library page of simulation, you can see the Synaptome model used, simulation configuration, and simulated results. You can also download the results. Let's also check our saved Synaptome model in the library. Go to the Models tab, click on Synaptome, and then click on your Synaptome model. This will open the Synaptome page showing its configuration and the simulations ran using the Synaptome model.